At the last part, we'll talk the actual cellular structures of bacterial cells. We are not getting into details in all these structures on the surface of the cells, but mainly focus on the bacterial flagellum and their swimming behavior. So what is a bacterial flagellum? It is a motility apparatus that bacteria used in their mobility. So a flagellum basically is a thin filament arranging from one or more locations on the surface of the cell wall. It is cellular locomotion, and it moves cells along this gradient of chemicals such as glucose or other nutrients. So there's many types of flagellar arrangements in bacterial cells. Here shows you the 4 basic types of flagellar arrangements in bacteria. The first one is monotrichous. So it is basically a single flagellum at one pole. So mono means one. In some cases, we also call it a polar flagellum because it's located in one end of the cells. And the second one is the amphitrichous. Amphi, it means two. It means there's a two flagellum located at the both ends of the cell. And the third one is lophotrichous. So lopho, it means bundled or the tuft of flagellum at one pole. And the last one is peritrichous, which is basically very common in E. coli and also in some other species. It means flagellum all over the cell surface. And besides the 4 basic types, there's also some bacteria that display some atypical flagellum. For example, in this slide, it shows you a periplasmic flagellum. Also, it could be called endoflagellum or axial filaments. So from the name endoflagellum or periplasmic flagellum, you can see that this structure is not displayed in the cell surface. Instead, in the next slide, you can see that it's actually displayed within the outer membrane. So it's in the periplasmic. That's why it's have these names. And in the next slide, it shows you the basic structure of the flagella of a flagellum. So it consists of 3 parts. So we will talk from the top to the end. So on the top, the yellow filaments shows you the real filaments that used and displays in the main flagellum filaments. It consists of protons, means named flagellin. And the blue part is called a hook. It is basically a universal joint connecting filaments, that connecting the filaments to the cells. And the larger parts, the yellowish combined with some reddish, this is the basal body of the flagellum. It consists of the motor that could rotate the cells, rotate the flagellum and move the cells swim towards. So this figure uses the gram-negative bacterium E. coli as an example. As you already know that the cell envelope of flagellum of gram-positive and gram-negative bacteria are different. So what's happened to the gram-positive bacteria? So you can see here, the difference basically located only in this basal body. In the negative bacteria, since they have this two-layer cell membrane, they have 4 rings located in this basal body, while in the gram-positive bacteria, they form these ring structures only in in this one ring cell membranes, and there's only 2 rings in this basal body. So this slide basically shows you 2 different types of flagellar models, and in details. In the left side, it shows the structure, the motors of E. coli. As you can see here, it is driven by the proton gradient. So basically, the respiration chance could make this proton gradient between the two sides of inner membrane, and this gradient could drive the ATPs and to produce some ATP, and also it can drive this motor in this basal body and try to have this flagella rotate. Well, E. coli, as you know, it's a kind of freshwater environment living species. So what's happened in this marine species, Vibrio? So this basically shows the basal body of Vibrio species. We call it sodium-driven polar flagellum. You can see the only difference between the 2 parts, it is in this motor part. So instead of mode A and mode B, the Vibrio display structures consist of POM A, POM B, which is homolog to the mode A and mode B. And also there's like a T ring, we call it. Actual T ring, it is mode XY proteins. Yeah. So, and also the important thing is the Vibrio polar flagellum, they use this sodium gradient instead of proton gradient. Well, these slides will introduce you a little bit about the assembling of flagellum. So the assembling flagellum, we call it self-assembling, and it's involved in more than 30 proteins. So first they start from this formation of the MS ring in the basal body. And after the formation of basal body, they will start this to form these 2 nanometer central channels in the center of the basal body. And through these central channels, the proteins for the hook or the end elements will be secreted out and try to self-assemble these filaments outside of the cells. So in these movies, we'll show you the details of the flagellum assembly. So it starts from the MS ring and the C ring in the basal body. Yeah. And now they try to form this central tubing things. And this is a cap for the hook elements. So the cap plays a function of the length determining. So now it's the hook part. When it's arrived the determined length, the cap will be released, and the new cap will be formed for this filaments assembling. It's very beautiful flower shapes. And now it's the white, it's the flagellum protons. And they try to form these filaments, in a very organized structures. And this process happens actually quickly in nature. And they will grow. Yeah. For each turn, there's around 6 flagellin protons. And between these flagellins, there's also some glycosylation, like some sugars will be added to modify this flagellin. So now you can see the surface of, the flagellar bundle display on this surface and help the bacterium to swim. Yes? So let's go back. And so the motors of the flagellum motors rotation lead to the flagellum rotation, and different flagellum rotation direction then lead to different, the two different motions in E. coli cells. When the flagellum rotates in a counterclockwise direction, so as you can see from the figure in the right part, the cells, when the flagellum rotates in a counterclockwise direction, the peritragus flagellum become bundled and push the cell swimming smoothly, or we can call this a cell run. However, when the flagellum rotates in a clockwise direction, they become choidic, the flagellum become choidic and separated with each other. Then the cells couldn't move smoothly anymore. So we call this motion tumble. And alternate between the two rotations will help the cells swim towards or away from the chemical gradient. Well, here also shows you films of E. coli cells with fluorescence flagellum, which is recorded by Howard Berg's lab. So you can see this, one of the cells, they are tumbling, the cells, and they start to swim. So maybe let's see again. Let's follow one cell, it swim, and then it comes to tumbling. So this is the swim, and then you can see the flagella just become separate, and the cells start to tumble. You can find more videos in their website, of course. And so what exactly control the cells to run or to tumble? So here, we use a terms named chemotaxis. So chemotaxis is actually achieved by a series of signal transduction pathway involving numbers of protons. You can see numbers of protons inside the cells. So the methylation and also the phosphorylation happens to this proton and makes a signal transduct from the receptor to the end of, and causing the rotation of the cells, of the flagellum. And it makes them counterclockwise or clockwise. Then you can see the real behavior of bacteria is run or tumble. So we will not get into the details because you will learn more in the future courses. Well, at last, I would like to show you a normal bacteria swimming that you could see with a microscope in the lab. of course, this video is to make you aware that without labels, you actually couldn't see any flagellum. So you can see the cells just swim very fast under the microscope. Oh, sorry. I just, whoops. Let's maybe try to send it. Yeah. So as you can see, there's no, you couldn't really see the flagellum of the bacteria, but you can still see this run and tumble behaviors of E. coli cells. And the different colors of cell body actually because the focus, now the focus just changed from one layer to the other. And yeah. Yes. Mm-hmm. Let's go back. So at last, it is a little summary of what we introduced in this section. So as the first part, we learned something about bacterial flagellum, basically the different type of flagellar arrangements in bacteria, and also some knowledge about the endoflagella or the periplasmate flagella. Also, there's like basic structures of bacteria flagellum filaments, which you really don't need to remember all these details, but you just know that the basic three parts of these filaments. In two slides, I show you the assembling of flagellum. And if you want to see more details of this assembling process, you can get to the YouTube link that's shown in the slides. And it's a half an hour minutes, half an hour films. And at last, I show you some swimming behaviors of E. coli cells. And there's some counterclockwise and clockwise flagellar rotations, and also the behavior of run and tumble motilities.